হ্যালো শুভদীপ, তুই ভিডিওটা অন করতে পারবে? ভিডিওটা অন হচ্ছে।
Sir, if you just on your video, you can do it. It is allowed. I have allowed actually. হ্যাঁ স্যার,
ভিডিওটা একটু অন করে দাও আমার। সেটা কিন্তু হচ্ছে না, বলছে হোস্ট হ্যাজ স্টপড ইট। হচ্ছে হচ্ছে Anyway, very good morning to all of you. On behalf of Jadavpur University, it's my pleasure to welcome you all in our webinar series. And this webinar is organized, it's actually a pre-conference webinar. Most of you perhaps know that we are going to organize the first, the ninth Asian Near Infrared Consortium in Kolkata. And this will be the first time this conference will be held in India. So as part of that, we are organizing a webinar series, and we started this series long time back. And today we have with us a very eminent person in the field of spectroscopy. He is none other than Dr. Rabi Narayan Sahoo. He is from ICAR, and he has worked extensively on different types of spectroscopy. And we requested him, and we are very glad that he has come all the way to Jadavpur University, and he's here with us, sitting just besides me. So we look forward to listen to his presentation. Before that, let me just give you a very brief introduction about Dr. Sahoo. Dr. Rabi Narayan Sahoo is working as principal scientist in the Division of Agricultural Physics, IARI, Indian Agricultural Research Institute of ICAR in New Delhi, and has more than 25 years of experience in research, teaching, and capacity building in remote sensing and GIS and its applications in agriculture. His major research interest is remote sensing applications in agriculture, hyperspectral remote sensing for soil and crop health monitoring, drone remote sensing, precision farming, plant phenomics, using sensors at ground, air, and satellite platforms. He has rich experience in generating and mobilizing resources to lead different multidisciplinary and multi-institutional national and international remote sensing research programs through different external funded projects. Dr. Sahoo developed state-of-the-art laboratories on hyperspectral remote sensing, drone remote sensing, big data analytics, and national facility on plant phenomics, and recently experimental learning center on drone robotics and machine learning, and 5G use lab for agriculture. Currently, he is leading ICAR network program on precision agriculture, NEPPA, involving multidisciplinary team from 16 ICAR institutes. Dr. Sahoo was also co-leading national network program on imaging spectroscopy and applications, NISA, of DST, having 37 research institutes covering different thematic domain areas of applications. He has published more than 240 publications, having 130 research papers in refereed and international and national journals, and mostly on remote sensing applications in agriculture. He has been an expert member to various committees of Niti Aayog, Ministry of Agriculture and Farmers Welfare, Department of Biotechnology and Department of Science and Technology and Indian Space Research Organization and ICAR Research Institutes. He was visiting faculty to University of Nebraska, USA, University of Saskatchewan, and Dalhousie University, Canada, and has been deputed to United Kingdom, Japan, China, and Luxembourg, USA, Thailand, and Canada for participation and deliberation on cutting-edge technologies for smart and precision farming. So on behalf of Jadavpur University and this conference committee of ANS 2024, we welcome Dr. Sahoo here at Jadavpur University, and I request to start your deliberation. I think all of us will be greatly will benefited from his lecture. So, Dr. Sahoo, please, the stage is yours.
Thank you, Professor Rajiv and his team, the organizers, for giving this opportunity to share our experience on spectroscopy and its applications. Are you able to hear me?
Yes, sir, we can hear you properly.
Okay, perfect. So I am putting my video off so that bandwidth will be better. Let me share my screen. Is it visible? Yes. Yeah. So good noon to all of you. Today, what I'll be talking about.
Sir, please share this thing in the full screen mode.
It is in the.
We can just see the part of the slide.
Are you able to see full screen mode?
Yes, 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 yes. Now it is good, sir.
Okay, just a minute. Okay, it's fine, no issue. So good afternoon to all of you. So today my deliberation topic is on socializing the pixel through spectroscopy, challenges and opportunities. Why I chose this topic is because I understand this is in the 6th in series, the talk is in terms of NIR spectroscopy. So I'll be talking starting from the non-imaging and imaging spectroscopy and its applications. So the title, Socializing the Pixel, means by how we can derive the informations which can be social benefit, better livelihood. That's what my next deliberations is going to be all through. And most of them are applications to the agriculture for the soils and plants. So look on the left-hand side. So what a human eye does with a biological sensor does is just a lateral image with a 2-dimension. But when you have a hyperspectral camera, you have a 3rd dimension where you have a number of spectral bands, which is over there. So click on any pixel, you are able to get a spectral signature. It may be 0.4 to 1.1 or 0.4 to 2.5 micrometer. It all depends the kind of imaging sensor what you have. And the resolution depends on the kind of the sensor's resolutions. It may be centimeter level, it may be meter level, it may be 30-meter level. So when you click any pixel, and that pixel has a spatiotemporal variability as the dynamic is the feature. And when you click on the pixel on the right-hand part, you can see each pixel speaks something about it. What is that pixel? What is that feature? So when you talk about the feature, for example, on the right-hand side, you click on a pixel, you are able to see the pattern of the soil spectra. Again, based on the pattern and up and down and the absorption and reflections, you can characterize soil and also its attributes. More detail I'm going to cover later. Same also when you look at the water body, you click on any pixel, you are able to see the water spectra. Again, you can characterize the all water attributes if at all you are going to analyze that spectral data. So same also with respect to vegetations and same also with respect to atmosphere. So what I mean to say is a pixel talks a lot about, provided you have a huge information, and that information comes from the spectroscopy. That information comes from the hyperspectral remote sensing. So this is one example I'm giving. This is borrowed from a colleague who has been working with us in this NASA JPL science team, Professor Robert Green, and we had a very ambitious program on average NC, which I'll be going to talk to you later. You can see the spectra, which covers from 400 to 2500. It has a radiance on the y-axis, and the x-axis you have the wavelength range, and you are able to see a spectra of a leaf of vegetations, where you are able to see the basic life, basic process of a life, that is photosynthesis, how wonderfully it is captured, both the right-hand side and left-hand side components. What I mean to say over here is, you can characterize the water, you can characterize the carbon dioxide, you can characterize the end product like carbohydrate and also oxygen. So you can also characterize how much photon get trapped based on the chlorophyll content. So what I mean to say is, you are not only understanding, quantifying the data, the parameter attribute, same time also you are understanding the processes. So remote sensing, the spectroscopy talks more beyond what qualitative remote sensing was doing till now, and also it captures more dynamic feature and also different processes on the Earth itself. So this is one of the live example what I am giving. Right-hand side in the dark table, what it tells you is what the physics spectroscopy happens, spectrometry happens. That leads to understanding the causes of observation, and also what the biochemical parameter attribute for that purpose. So that tells you, if at all you have a spectral signature of a feature of a pixel, you can characterize how that feature is behaving, what are the compositions, and how it is going to be throughout its life cycle. So that's what the wonderful gift with respect to spectroscopy when you look at. Coming back to why we are talking of spectroscopy or hyperspectral remote sensing, when you compare with the conventional remote sensing till now, what we had is a multispectral remote sensing, where only you have few channels. For example, on the left-hand side, you can see the spectral signatures of three major features of the Earth, that is soil, water, and vegetations. But when you look at most of the satellites which is covering in the multispectral, in the spectral range of around VNR, RGB visible range, few bands to 32 or 40 bands. For example, look at the Moody's, look at the Sentinel series data. So you have a discrete feature for that purpose. Right-hand side, it shows that. So you have a discrete feature, the capability of discriminating or characterizing that feature is going to be limited. Same time, when you look at a hyperspectral sensor, either from the ground or from the satellite, when you click on any single pixel, you are able to see the continuous spectra moving from 400 to 2500, and you'll be able to get the features in between which can be characterized. The basic process of remote sensing is nothing but the function of biological, physical, and chemical attributes. So if at all each part of the spectrum is captured, that means you're capturing all the information related to the target is going to be rich enough to characterize that target or more understanding of that. So look at the soil spectra also here. That's a mineral which is called kaolinite. You can see the multispectral feature, the spectral signature of multispectral, that is the TM band. And look at the hyperspectral data characterized in the lab conditions. You can see how wonderfully up and down is there. So discrimination of this kaolinite mineral from others and understanding that why it is behaving like that with respect to hydroxyl bonds or any other chemical changes will be more conspicuous, more visible if at all you have a hyperspectral data. Same also, and you can understand the physics behind E, if ever at all you have a spectral range starting from 0.4 to 2 micrometer, each spectral range is characterized or is governed by a number of factors, which is more of a physics and also physiological processes. For example, initial part is more governed by pigments, next part is internal reflection, next part is the water, lignin, cellulose, like that. So each part corresponds to something ground on the vegetation or on the soil or on the water. So if at all I characterize that in more in detail, then that means I'll be able to understand better the ecosystem I am living in and better management options I can. So that's what I'm going to talk more on detail on that only. So looking at the sensors, what we have till now is we have a ground-based both imaging and non-imaging sensors. We have airborne sensors. There are a couple of them are given over here. And most important and most visible sensor all worldwide applications people have been referring to is JPL NASA average sensor, which is given over here. And then you have many satellite-based sensors right now, which is live is Prisma and Nmapper from Europeans. And you look at the ground sensors, there are many imaging sensors which can be also mounted on the airborne platform, and you can capture. And you can see, starting from the lab conditions, using a non-imaging spectrometer, you can characterize a soil in a petri dish to see how wonderfully you can characterize with respect to quantitative analysis of the soil fertility attributes or any other parameter. Same time also, you can go to the field, see what are the noises, what are the practical challenges, what we have in the field conditions. And again, when you go to the airborne or satellite platform, altogether the story is going to be different. So purpose of doing all this possible scale is how we can make it scale neutral using this kind of technology for different applications, particularly for agriculture with respect to soil and crop health monitoring. That's what the purpose over here. And you can look at the number of sensors. If you see, they can be clustered into 3 vertices. The sensor which just give the spatial information, those are the images. For example, you have mobile camera. The sensor which gives the spectral information, that is spectrometer. You have many spectrometers in the lab conditions. And you have also a single channel radiometer which measures the intensity. You take all possible combinations of these 3 vertices, the triangle, what I am saying. You take the spatial attribute and you also take spectral attribute. That pulling them together is called imaging spectrometer. When you talk of spatial and radiometer, that becomes imaging radiometer. When you talk of spectra and intensity, that becomes spectroradiometer. When you pull them together, that is imaging spectrometer. So the challenge remains is how from the non-imaging radiometer and the lab conditions to the imaging spectrometer, which is on the airborne or satellite, we can use for different applications. That's what my deliberation is going to be now. And you can see these are what possible platforms and the sensors what we have in the lab and possible protocols what we developed over the time, and try to evaluate all those sensors to see how best we can use for the soil and crop health monitoring in the different conditions. And we have also ground conditions in the climate control conditions, where we have 2 imaging sensors, both in VNR range and SUI ranges, where we are able to capture at a go around 1,200 plants in a pot, which is climate control and differential treated with respect to nitrogen, water, or any kind of treatment you can give, and see the genotypic response, how they behave, and characterizing them with using this VNR and SUI hyperspectral camera, which acts as a PET scanner for the plant, and you can characterize them more better than any other single channel camera, for example, NIR or thermal camera. Same sensors also we have on the drone and also on the mobile platform, where you can move in possible directions. So, and coming back to the remote sensing, long back in 1969, we had a black and white picture, and the first initial study started with the coconut oil disease in Kerala. And over the 55 years, we have moved through, starting from ectachrome camera now to hyperspectral camera with a high resolution radiometric spectral resolutions. And that's what the advancement has been taken place. So what we could do that time is just simply slicing the image and to see and correlate with the ground truth to see how coconut root wilt can be discriminated. But right now, same place, again, we have flown in 2019 with the hyperspectral camera, not only to see the coconut wilt disease, also discriminate different diseases and also disease severity level. So that's what the technology advancement has been taking place. And this is what we are looking forward. And if you look at the aerial survey in India, this is what happened in 1969, and very recent, what we had is with NASA ISRO collaborations, we have imaging spectrometers like average NG. And now also in October this year, we are going to have also science campaign in many parts of the country to see how best whatever the technology we developed can be used in the large scale for different applications, so that India can plan for a mission on hyperspectral camera for the civilian use. And you can see the number of initiatives which has been taken over here, like airborne high-sea data on the helicopter, which was taken care by ISRO. And we had ISRO JPL NASA program, where we have flown different part of the country, around 55 sites we have taken with a different thematic area cover, around 2016 to 18. Again, in 2024, we are going to have it. Again, we had a very ambitious program called Network Program on Imaging Spectroscopy, covering almost 7 themes and 37 collaborating institutes, looking at the possible application of both imaging and non-imaging spectroscopy. And so again, IIRE has recently started many non-imaging and imaging data acquisitions and from the different platforms for precision agriculture. Looking at the hyperspectral imaging sensors in India, again, you go back to 1975. Till now, India has been trying to see if at all we can develop different sensors, and they have been launched at different time scale at the airborne and also on the satellite and also on Chandrayaan missions. So this is what the average energy science campaign we had. This is what the science team with the JPL NASA, and these are the sites where we have visited with having our ground non-imaging sensors. Same time, we have flight borrowed from ISRO and integrated this NASA sensor, and there's almost 100 kg and flown a different part of the country, and try to see how this hyperspectral or spectroscopy can be used for different applications in the country. And you can see the kind of data what is covered around 3 to 8 kilometer height and around 425 spectral bands. And with mercury cadmium photodetector, which is used there in the imaging sensor and integrated with a King Airbus 200 of NASA in the ISRO flight. And same time, we are on the ground collecting the data. And this is what the data has been captured, at least where the IIRE was involved with around more than 14 sites, collecting different features, different soil, plant, water, and many other attributes. And coming back to the satellite, there are many satellites, of course, right now it is available, but long back, Hyperion satellite was there, which was a hyperspectral sensor having around 242 channels, but finally taking out all the noisy bands, what atmospheric noises are, only 158 bands were there to be used with a 10 nanometer bandwidth, and that kind of study was done. And after that, of course, we have now, right now, Prisma and Nmapper, which is available, anybody can use. So you can look at the ground non-imaging spectrometer data, which is written there, ASD is sieved CP. CP is the contact probe, and ASD is the instrumental spectrometer, and sieved is the soil, which was processed through some 2 mm sieve mesh, and that has, this is what the spectra you are able to see, the blue color. And when you same petri dish you took out and do in the sunlight conditions, how the spectra is changing, that is what the red color. When you go to the field conditions, the down one, you can see the lot of noise is coming. And those blue narrow width, what we have given is those are all the noise bands because of the atmospheric interactions. And last one is the Hyperion image. The right-hand side is the average energy image. You can see the pattern, of course, remains same, but there are a lot of noises you can see when you go from a single pixel in the lab conditions to the field conditions having more than 10 by 10 meter, 30 by 30 meter, a lot of heterogeneity, a lot of noises because of the atmosphere. Again, the amplitude keeps on increasing when you go from the satellite down to the Earth because the signal noise ratio is going to be much better. You can see on the right-hand side, you have a satellite data that is Hyperion, which is the blue one, and average energy, which is 3 to 8 kilometer height, which was flown, that is a green spectra. And the ASD spectra, which collected in the lab conditions, hardly there is any noise. You can see the amplitude all through is very high. So that's what the kind of data what you have. And the challenge remains is how from the ground non-imaging spectrometer, we can scale up to large scale for different applications where a lot of challenges are signal noise ratio and a lot of noises because of the atmosphere, water vapor, and many other attributes. So coming back to the applications, mostly because I am from the domain, I'll be covering most of the applications related to agriculture and related to the soil, how we can characterize the soils, map them, soil fertility and quality monitoring, land regressions. Coming back to the plant, the biotic and non-biotic stresses like nitrogen, phosphorus, potassium, sulfur, water stress, biotic stresses like insect, pest, and diseases, a new concept of a hyper BRDF, bidirectional reflectance, and also ready to transfer modeling, how we can use for. And of course, because of the time constraint, I have taken out the post-harvest quality and grading part right now. I didn't keep over here. And these are the different approaches we try to use, simply univariate approach or the spectral gradage or empirical modeling or physical modeling or machine learning models. So coming back to the program, I was co-leading the National Program on Imaging Spectroscopy and Applications, where we had 37 sub-projects, and we had 4 central lab facilities all around the country. And you can see the kind of the 74 PIs and co-PIs with a good spectroscopy community. We had around 148 people who are working together. This is what the team, which was launched in the IIR campus by the 2 secretaries, one from the DARES and also from the DST. And this is what the team, this is a boon for us to get the free data what we had. And thanks to the ISRO, they shared all the data with us. And this is what the delivery finally we got. These are the centers what we have, and this is what the thematic applications, starting from geology, agriculture, water, forestry, snow, ice, urban built-up, and algorithms. And we have almost 34 students who did their PhD on imaging spectroscopy. And you can see the 126 publications. This is a 2 years back status right now. I have not updated, of course. So this is what the kind of work what we did in the last 5 years on that. Coming back to some of those applications, you can see starting from the ground to airborne hyperspectral sensors, you can see the spectral data of a soil when you take in the lab conditions. And this is what the 400 to 2500 range, you have a reflectance data. And when you look at the APR, which works from 2 to 16 micrometer, and that's the wave number is 4000 to 500, you can see the observance spectra, how it behaves. And at the same time also, we have the instrument which has emissivity, a measurement also, you can see the emissivity. And same time, you have a average energy sensor, which was flown up on the same soils, and you can see the data which has been captured. In between, you are able to see those gaps because of the atmospheric noise. Those gaps have been removed. All this data which is collected over here, you have to see, those are all the lab conditions. That's why you don't have atmosphere. That's why you are able to see continuous spectra. When you go to the average energy, so we have taken out at 1400, 1900, all those bands, because those are atmospheric noisy bands. So we have removed those to do equivalent analysis and comparisons. So we try to do that visible NIR spectroscopy and MIR spectroscopy. We try to pull them together. You can see the spectra, which is collected using the spectrometer from 400 to 2500, and the MIR spectra, which is collected from the FTR using 2 to 16 micrometer. We combine them together to see the spectra starting from 400 to 16,400. This is what the spectra of a soil, how it behaves like. This is a wonderful library one can characterize and do it. So, and at a go, you can go for processing, data mining, all kind of big data analytics you can do using different approaches. And those approaches are like given here. So you have a major spectra, that's a reflectance, and you can convert that into first derivative, second derivative of reflectance and also observance. And again, you can go for the spectral pre-processing, like noise removal, spectral smoothing, and data normalization, removing outliers. These are all basic steps what usually we do when you collect the ground spectra. When you go for the airborne spectra, of course, we convert those raw digital number to reflectance and go for the atmospheric corrections and remove all those noise bands, and again, spectral smoothing, image mosaicing, data normalizations, removing outliers. And this is what the number of soil parameters we try to approach. And these are different models. Of course, we have gone beyond these models. There are different machine learning models we try to do. And there are different feature selection techniques. You can see which band is very sensitive for the organic carbon, which is good for nitrogen, which is for potassium, phosphorus, sulfur. Each individual attribute can be done using the feature selection procedures, which is given here, starting from ANN, artificial neural network, to variable input and projections, Mars, random forest, PLS like factor loadings, and then cars, PLS. Like this, there are many other techniques that are available. And if anybody is interested, we can discuss more in detail. Coming back to the analysis, intersensory evaluation, starting from the ground measured VNR spectra, that's the VNR ASD. ASD is the company name, Analytical Spectral Devices of US. That's what the spectrometer which captures 400, 2500. Similar data you have captured on the average energy. And we try to do, what we did is, whatever the average energy spectra we had, we took the average energy sensor response function, we try to integrate whatever the ground collected data we have from the spectrometer. And ground collected spectral data is at 1 nanometer bandwidth, whereas average energy is something around 5 nanometer bandwidth. We try to integrate that using sensor response function of average energy. So whatever the spectra you are now simulating from the spectrometer, that is going to be equivalent spectra of average energy, not at 3 kilometers, but it at on the ground, so that you can go for intersensory evaluations. That's what the standard protocol usually global people follow, and we try to follow the same. And we also try to evaluate with respect to MIR observance and MIR emissivity, and try to combine both visible NIR and MIR spectrometer data to see which sensor is really good with respect to predicting all these soil attributes. You could see those bars with respect to the calibration and validation and the prediction equation R square. You could see most of the time it is the MIR spectroscopy which looks to be much better than BNR spectroscopy, and that goes good with a good accuracy. Next follows is visible near infrared spectroscopy. Next is BNR and emissivity. And finally, the most lowest one is the BNR average energy, which flies something around 3 to 8 kilometer height. And obviously that is true because the resolution is going to be 3 by 3 meter, and you have somewhere around 3 kilometer height. There are a lot of noises. You are losing some information in some of those very important bands like 14 and 19 and there are a lot of heterogeneity with respect to 3 meter or 8 meter by 8 meter pixel when you look at. So that's the reason accuracy has gone down. But nevertheless, that can be used also to go for the kind of prediction. Is most of them you can see around more than 0.5. So you can go for the significant analysis. You can see how wonderful it is. So keeping that in view, we try to do the predictions using both the airborne imaging spectrometer and also lab-based spectroscopy, and develop all those soil fertility map simply using different machine learning models. This is what, for example, you can see. This is somewhere in Karnataka where you are able to see the natural color composite taking 3 bands from the hyperspectral data, which is collected from the average NG. And we classified again using a hyperspectral classifier like taking MNF and all those things. Then finally, we got the soil area calculations masking out. Then the again carbon map you are able to see. So what I mean to say is you have a raw image with a pixel. You don't know what is covered by, but at the end, when you get the spectral data, you know what is the feature is, whether it is a naked soil or it's covered with some crops. Then going for the spectral angle mapper, all kind of a hyperspectral classification technique, you try to classify those crops and try to remove them out, get the soil part, and also characterizing the organic carbon. So this is what the socializing the pixel. When you go from one pixel to another pixel, you are able to see the variations with respect to organic carbon content. And today, maybe another 2 years time, 3 years time, you will see what is the change. So this is what that pixel you are able to characterize with respect to the intervention taken on that pixel itself. So you can see that same pixel, you look at the naked eye, that looks simply brown color. When you scan through average energy, this is what the wonderful color you are able to see. That tells you there is a variations with respect to organic carbon or fertility attributes. Then you can go for variable rate applications. So this is what example. Then this is what the airborne data analysis. Same time we try to do also both on the lab conditions, field conditions, and also satellite level analysis. This is a study done long back in 2008 when Hyperion was active. This is somewhere in Jalandhar area. And you can see the prediction parameter with respect to NPK, soil organic carbon, calcium carbonate, and also texture. So we try to do, again, you can see the similar kind of studies. You could see lab condition. Of course, the prediction model will be always good because there is no noise. And when you go to the field, there is always a noise. Never soil is exposed as pure as in the petri dish in the lab conditions. That's the reason why the prediction model has gone down. When you go to the satellite data, there is a 30 by 30 meter resolutions, pixel, lot of heterogeneity, again, noises, and it has gone down. So this is what we try to work out, how we can scale up and what are the challenges we have that one has to address to bring the further improvement on those R-square prediction model. So keeping that in view, we have been collecting a large-scale spectral database and trying to see how best we can use them for deriving soil fertility attributes, non-invasive and rapid, and that too in a special scale. So this is what you can look at the soil health card program, what Government of India is having. So there are two approach we have for that. One is, if at all you get the soil to the lab conditions and you put the spectrometer over the soil, which is on the petri dish, within no time, we could get the prediction done with respect to available nitrogen, available phosphorus, available potash, and organic carbon. This is the major nutrient what we always look for. Based on that, only recommendation of fertilizer is given. And this is what the spectra collect on the right-hand side. And based on that, already it is inbuilt in the my system itself. You are able to get what is the available nitrogen, phosphorus, and potassium. That's what the green chart you are able to see. So we are trying to customize, making Indianized to see how best we can come out with the sensor. You can see, you take the sensor in the field and capture the data. You should be able to get the soil fertility attribute within no time. We are trying to improve the accuracy further, though it is right now, it's not that good, though it is 0.6 or 0.7. But for this purpose, we need to have a large data set to work, covering all kind of 10 soil orders what India is having, covering all kind of challenges what we have with respect to soil variations and attributes. Coming back to the next concept is the digital soil mapping for again soil health. When you look at how digital soil mapping is nothing but which again takes care of hyperspectral data and also some of the environmental covariates which affect the soil formations, pulling them together, running machine learning. We try to derive a spatial map of water state of the organic carbon, available phosphorus, and potassium. So each pixel you click, you will be getting what is that organic carbon content, what is the phosphorus content, what is the potassium content. So you don't need to do nitrogen because organic carbon and nitrogen has a good relationship. That's the reason once you get the organic carbon content, indirectly you know the nitrogen content. So this is what the socializing, familiarizing the pixel. In that pixel you click, you are able to get the soil fertility status. Then if at all you are growing a crop over there, then what to be done, what not to be done, that kind of advisory can be generated. This is what the similar concept we try to use for the whole IRE farm, which is a very big, around 323 hectares, around 19 soil attributes we have. And this is what you are able to see. The beauty of this digital soil mapping concept is very dynamic. You don't need to invest any money on this. So you have all satellite-derived indicators. You are trying to pull them together. And again, we have some legacy data of the soil and also spectra, pulling them together and characterizing this and validating that in the field. So the legacy data, what you have here, for example, we have 282 data points. We try to validate how good is your data point for that purpose. You can see the R-square and concordance, how good it is. So this is what you can do also in the large scale to get the soil fertility, but the accuracy is not that good. If at all you can improve upon further the accuracy, that means you can see how wonderful it will be a contribution to the digital soil health card program of the country. So you get into the lab, complement to the weight chemistry through the sensor what you have. That sensor cost is around 80 lakhs or 100 lakhs, but it doesn't matter. We can see how much money we invest on the soil weight chemistry. And of course, leaving aside that also, but that will be much better to do because the accuracy is much better. But when you go to the field scale like this, you click on any pixel or you have a mobile, you have a locations there, you stand over there, within no time you click, you'll get all attributes on your mobile itself. And of course, the accuracy will not that good what you have in the lab conditions. So, but it is always you have a better idea about what your soil fertility is and what should be done. So this is what, again, we are trying to work with many institutes to be successful. So this is what I was talking to, again, socializing the pixel. So this is what the digital soil map of a district of Madhya Pradesh, Dhamantari, where, so you are able to get the nitrogen, phosphorus, and potassium map. So the farmer is standing there in the field itself. He is able to get that data. This is a mobile app which has been generated. And the server has the digital data, soil map data. And from there, he's able to look at his field. He's able to choose his crop, what it should be, and how much fertilizer to be added, that he can get the recommendations out of it. So this is what we are doing, socializing the pixel with respect to what kind of management options he should choose for, only based on the soil spectroscopy and soil fertility mapping. Coming back to the analysis, what we are trying to do right now is we have the opportunity to have a free data available on the Prisma and the Nmapper. For example, this is what we requested. We got the data acquisitions very recently. And you can see the kind of data sampling which has been done in 2 fields, around 200 sample points we have taken. And these are the attributes what we have analyzing. And this is what the ground collected spectrometer data you can see, ASD spectrometer data. We have 2 cameras, which is 400 to 1000 nanometer and also another 400 to 2500. This is imaging spectrometer, which goes on the drone and flying at the same time. So you have a satellite data available, for example, those ET-lens satellite like Prisma and also Nmapper. And same time we have a ground data, same time we have a airborne data to see how wonderfully that can be connected, starting from a single pixel to a large scale for the soil fertility mapping. So this is what all about the soil. Coming back to the crop, we try to see also how how better is the spectroscopy compared to RGB or multispectral. As at the initial slide, I talked about multispectral vis-a-vis hyperspectral. You can see over here, RGB has only 3 bands, whereas you could look at the multispectral band, which is in the drone, we have around 5 bands. Those red one is going through. And when you have a hyperspectral, you can see how narrow width, you have a lot of data collected over here. So that's what the beauty of data collections and huge data. And any kind of attribute you want to characterize with respect to plant or soil, that's possible on that. And this is what the field with the differential treatment of the nitrogen and irrigations. This is what the drone with the camera flying over there, which is from 400 to 2500, having 600 channels at a go. So each pixel you click, you will get a 600 bands pulling over there, spectral signatures. And this is what the applied soil nitrogen treatment, and this is what the measured Li and measured nitrogen, leaf nitrogen content, which has been captured in the ground itself through the wet chemistry, through the conventional approach. And when we try to do the best on the ground spectrometer or the airborne hyperspectral data from something around 20 to 25 meter height, we have flown. And you can see the kind of spectra which has been generated over here on the left and the right hand side after doing pre-processing. These are all machine learning models we try to do. How ground spectrometer is good with respect to nitrogen estimations, how airborne is good with respect to nitrogen estimations. So similar kind of results you are able to get with, and you can see how good it is matching with respect to that. And you can see the kind of validations. These are all published work. Always I can share these publications. And you can see how leaf area index, for example, the first one, how the leaf area index is a indicator, growth indicator of a crop. And also most of the time people enforce Li for the yield estimations and also growth monitoring. And the next one is the LCC, is the leaf color, leaf chlorophyll content. And last one is canopy chlorophyll content. And how the spectra is changing, if the change of Li, change of chlorophyll content, change of the canopy chlorophyll content. And this is how, based on that, we try to learn all those machine learning models to generate this thematic product of the field. And that turnaround time is around only 1 to 2 hours time. So now the question is, you look at the picture. This is a raw image on the left-hand side. This is a data captured from the drone. And each pixel you click, this is what the spectra, and that tells you the treatment, what you are given in the field itself with respect to irrigation and nitrogen. So nitrogen is starting from 0 to 240, around 8 treatments was given. And finally, after processing, you can see the color map tells you the nitrogen concentrations. So from a raw image to thematic product, you have generated a nitrogen variable map has been generated. So in a field, that variations you are able to capture, not only from plot-wise, also within the plot, each pixel, the nitrogen content variation is there. So what I mean to say is how this pixel is based on the spectroscopy, you are getting that rich information. Now I have to go for the management. This question lies, my management decision should be what? What based on the nitrogen, what I should apply, that's what the management options I should generate based on the distance support system or whatever the expertise you have, then go and apply that. This is what the precision agriculture, this is what the variable data applications. So now what people have been doing is like drone-based applications of pesticides or herbicides or nutrient application they're doing. Unless until you go for the understanding where it is a requirement, why you should go for uniform spraying or uniform applications. So this is what the solutions should be. It is always a precursor for any kind of input applications to know exactly how much is required at what pixel. So this is what I mean to say, from a raw data to a thematic management option data, you are able to do because of the spectroscopy. Coming back to the latest advance, that's what the bidirectional reference distribution function. What angle you should look at to see or characterize a feature, that's very interesting. Very nice. Till now, since till now we have most of the sensors were in the Nadir. They're capturing almost semi-spectral data. But there are many, like Modis, you can look at, and there are many sensors are there, which are on the BRDF sensors. BRDF is in the different directions. Bidirectional reference distribution function it is because it has two directions. One direction, the radiation is coming from the sun, and another direction you are receiving from the sensor. So there is always in a particular direction, the radiation is going to be different, because always the radiation which is going out of the target from the Earth, they are non-isotropy in nature. That means in any directions, the radiation is not going to be same. It is not equal. So since it is a non-isotropicity, so you have to look at a particular directions, at what directions the real radiation is getting maximum, and we can retrieve more information. So for that directional property of the target to take care, this BRDF sensor has been developed. This is what our in-house developed the sensor called the kind of a facility called goniometer, which we try to use in the different zenith and azimuth angle. Sensor can move and capture the data. So you know sun's positions, you can calculate the sun's position based on the locations and the time. So solar geometry is calculated, and then based on that, you can calculate also your sensor geometry. Then relatively, you can calculate how good or bad is your position with the radiations with respect to the sun. So for example, you can give an example here. You look at the soil, same soil, the radiation is getting different because of the different angle of zenith. My relative azimuth is 0. Relative azimuth 0 means the azimuth of sun and azimuth of my sensor is same. So relative means if I minus it, that becomes 0. So I have taken relative azimuth 0, only the zenith I am changing. You look at the soil spectra, how it is behaving. So that shows you how isotropicity, non-isotropicity plays a big role with respect to characterizing any data. So that's the reason always there should be SOP, there should be standardized protocol, what angle at what to look at the target so that you can characterize the information much better. Same time, you can look at the crop. This is a wheat crop. Again, same crop you are looking at, but the spectra is getting different. Now, next question is, so you could see the non-isotropicity because of the spectra is changing, though the soil remains same or the crop remains same. That's one. Second is you can see the maximum value you are getting somewhere. You may not be able to visualize it. Sorry for that. Maybe something around 40 degree, you are able to see that value. The 40 degree is the maximum, the radiation which is going on the topmost is the 40 degree. Why it is 40 degree? You look at the sun zenith, that is something around 46. Since I was taking a 10 degree interval, so that means where the sun's zenith position, same zenith position sensor should be, so that you can get, that's called hotspot effect. For example, you do the photography. So when you do the photography, you do always putting the sun on your behind. So same concept also is here naturally. So same also with respect to wheat. So this is called hotspot effect. And based on that, this property plays a very important role with respect to 3D simulations of any feature. That's in defense, we do a lot. And also in case of agriculture, we can go for the phylotaxy or morphometric analysis of plant. One is a small dwarf variety, another is a tall variety. You can discriminate them using simply non-isotropicity and all these properties. So because of the time constraint, I'm not going to more in detail into that. Second is, till now what I was talking about, the empirical modeling or the simply machine learning models and deep learning. And you understand the limitations of those empirical modelings. So again, the developments recently is in physical model approach called radiative transfer model approach. That means how the radiation comes, interacts with the plant, goes down to the soil, again comes back, interact with the canopy, goes back to the sensor. Each part is modeled, understanding the physics, chemistry, and also physiology behind it. How the plant, the radiation is coming, how it interacts with the plant, that depends on the plant attributes. As I said at the beginning, chlorophyll content or water content or internal reflections, cell structure or transit conditions. Then it goes back to the soil, again come back. So they are all governed by all the physics behind it. So those can be modeled, and that's what called radiative transfer model. And you can see people have been doing 6x model, people have been using for atmospheric modeling, all atmospheric condition studies, like you see atmospheric scientists, they work for that. For agriculture domain or plant science, you can look at the canopy structure, leaf area index, leaf angle distributions, all those things, leaf chlorophyll, water. This affects your radiations. Then also soil variables you can capture. Then you can, this is what the radiative transfer model, you can simulate a spectra based on that. So this is what called radiative transfer model, forward modeling. So understanding that, so you put the input into the parameter, your function, you can get a simulation spectra like this. So like this, you can create a million spectra. I'll come back to that later. So this spectra you can create and generate put in a library in your server itself. Million spectra you can generate as the field conditions varies. Lab condition, it doesn't need any money. Same time, you have a measured spectra, either from a ground spectrometer or from a imaging spectrometer. Then you have to go for reverting it. So you had a Y is your reflectance is a function of all those attributes. Now, I want those attributes based on what reflectance I have. So now what I need is that attribute is equal to F inverse the reflectance of the spectrum, whatever the spectral reflectance or observance or whatever data you have. So this is what the inverse model. So this is what the spectral measured data I have. Then I go for this is what the measured spectra. I go for inverse modeling. Again, inverse modeling, there are different approaches one can use for and can retrieve these parameters. And how about those parameters? You have also ground measured those parameters like leaf area index or chlorophyll. You have a ground conventional approach measurement. You got a predicted and you can always go for accuracy assessment. So there is a forward model simulating the spectra and optimizing that model so that whatever the measured spectra and simulated spectra, they are matching. That means your model is perfect now. Then you have a measured spectra now. You can invert that model to get the required parameter what you look for. So this is what the radiative transfer model in just in a brief summary. Again, because of the time constraint, I can't stay over here. So this is what the kind of radiative transfer model, and these are the different parameters which you measure. For example, leaf structure, chlorophyll, equivalent water thickness, dry matter content. And this is a very famous model called ProCell. Both Cell and Prospect, they take pulling them together. These are two scientists, they worked on it and combined them together called ProCell model. Prospect takes care of most of the physical attributes and Cell takes care of most of the angle sensor attributes. And pulling them together, you can simulate the spectra. I'll come back to that later now. So these are all around 14 parameters based on which you can simulate the spectra. This is what the forward modeling can do. And you can measure with the observed spectra, you can compare and go for the evaluations of them. Let me tell you one example. You see here, this is what the chlorophyll, this is what the model is. Inside I have model. I just keep on changing the chlorophyll content and you can see how the spectra simulation. This is around 1,000 spectra which is simulated. And you know the basic spectroscopy of the plant, the chlorophyll only makes the change in the visible range, not beyond that. And this is what you are able to see over here. Beyond that, only a single is going. That means perfectly they're matching. So the sensitive part of the spectrum for chlorophyll is, this is the range, 400 to 700. So you are able to see they capture that. Going to the next, this is what the water content. This is the sensitive band of water content. Again, you go to the spectroscopy, you know how the water content changes and how NIR and MIR part is very sensitive for the water. And you are able to see the spectra changing. And this is what the sensitive band. So this is what the sensitive analysis of the simulation of spectra you can do based on the plant attributes. Those are water content, chlorophyll, and a leaf area index. Next one is the leaf area index. So like this, you have n number of attributes. You can do the sensitive analysis. Once it is done, you put into the model and run the simulations. So you know, starting from 0 to 10 is the LI value at every 0.001 interval you can do. All possible LI, you can go for chlorophyll. All possible chlorophyll, you can go for water content. So you can create million spectra and save in your server. And this is what the simulated spectra, exactly what it can be when you go to the ground and all scenario you have built up. So once you have created, that's called lookup table. Once you have created the million spectra in the server, you have a major spectra from a single pixel. That pixel, that spectra goes matching with this million spectra. Where it matches, that matching itself is a cumbersome job. That's why we look for the machine learning models. There are a lot of publications I can share with. Then it retrieves the parameter, corresponding parameter of what it is. So this is what the radiative transfer model. And once it is developed, that's going to be generic. It don't change with the time or from place to place because there is no coefficient variation. There is no subjective. It's a process-based model. This is what the million spectra you can creating and generating that. And this is what the end product you can see. This is from the drone we have done because of the time constraint. We have done also with the satellite with the large scale operations. And now we are making make it more of operation. This is what the LI product generate from the same radiative transfer model. This is what the canopy chlorophyll content, nitrogen content. Finally, pulling them together, you can go for the grain yield because these are the major attributes for the grain yield. and you can go for the granular estimations for that purpose. So this is what I talked about the nitrogen and chlorophyll and water. Coming back to the biotic part, these are again hyperspectral plays an important role with respect to biotic stresses like pest and diseases. And you see, for example, you look at the brown plant hopper. Again, there are a lot of challenges with respect to pest and disease. I am not discussing those things right now here, but you are able to see the spectral variations with respect to the scoring, which is done from 0 to 9, and how they vary and which parts are very sensitive bands. You can see those bars over here. And when you go for the simply correlation analysis, you are able to see how NIR band is very sensitive with respect to the high correlations, and those bands are very sensitive for the BPH monitoring and also predicting the scoring. And this is what the prediction which has been done. We have challenges for that, but now I am not discussing with respect to that. Coming back to the wheat yellow rust, again, you can see the spectral variations with respect to the scoring which has been done. You are able to see the NIR spectra data is a lot of variations. Are you able to see here when you go from 700 all of a sudden 680 to 780, it almost goes as a single line. But when you take the derivative, you are able to see wonderfully they are differentiated. How the slope changes, that's what called the red edge technique, red edge approach. So, and that red edge approach is possible only when you have a hyperspectral data, because this shifting of the peak or the amplitude that is the parameter which can be used for the nitrogen stress and also pest and disease. And that shifting is going to be only nanometer interval. So in multispectral, red edge cannot be useful. Only in hyperspectral we can use. And looking at the importance of this, the new sensor has come up in the drone. If anybody is using drone remote sensing, there is a multispectral camera which has a red edge band, MegaSense red edge band, Parrot Sequoia with the red edge band. So red edge is this is why it is called red edge. From the red to NIR band, when you go, and that edge, there is a point of inflections, and that point of inflection is the red edge point. So, and you are able to see that derivative, and you see the prediction model how it works for with respect to yellow rust. This is a very recent publication work which we did. Same also with respect to wheat yellow rust from the ground and also from the satellite data, average NG data, which has been done. So again, this is what the wheat feed, again using spectroscopy. I'm not going to talk in detail again. 20 days after inoculation, 40 days after inoculation, how the plant is behaving, how that pixel, that plot is behaving, you are able to see that. There are other treatments, everything remains same, only the infection was there. So whatever the changes is because of the aphid populations. And same thing we are trying to do from the drone also. This is what the rice blast, again, one of the important disease in case of rice. We try to do, again, the spectroscopy, we go for the derivative. As I said, red is when you go for the first derivative, when you go for a distance versus time, when you go for derivative, you know your velocity. When you take velocity and versus time, you get the accelerations. So always derivative gives something else, whether you have uniform velocity or uniform acceleration or you are a static. So same also when you derive the spectra, how the spectra is changing because of little change in the processes there, that can be magnified. You are able to see a lot of peaks up and down. This is what the correlation coefficients, and these are mostly at the NIR ranges you are able to see. These are very sensitive bands which we could identify for the rice blast, which is very useful. And not only that, we had a 9 scoring we have done, and we try to discriminate also scoring based on the hyperspectral data. So not only you are able to do the disease mapping, also you are able to discriminate the stress levels of the diseases which is happening. For example, 1 to 9. And again, I'm not going to talk the detail of that. The value which is come, that's called spectral distance we calculate based on some calculations. And you are able to see that's a red dot which tells the stress level 4 and 5, they cannot be discriminated. They look same. So this is what the challenges what you have. And then we try to run the models to predict the severity level. And then I think I should skip. I don't know how much time I have. I think I have an extra time, maybe another 1 minute, 2 minutes. So coming back to the early prediction of the pest and disease, this is a big challenge again, which is coming up. How you can do that, and the spectroscopy can be helpful for that purpose, because whatever the biochemical changes takes place in the plant, that can be reflected in the spectral data starting from 400, 2500, beyond that if you are looking for. So we try to do those kind of analysis. Again, to explain this, it takes time to, this is what the spectral contour plotting to see the day 1, day 2, day 3. When I go for the lambda plotting, I will be able to see some changes over there. That means there is something happening on the ground itself. Those are the plant defense proteins, pathogen-related proteins, and protein expression or regulations, which plant inside is happening. And that is reflected in my results. And I am able to identify, this is what the sensitive bands, that is NIR band around 1064 and 1300, which is very sensitive for early predictions of the rice blast. So, and again, understanding the physiology behind it, what are those defense mechanisms behind it. So this is what the spectroscopy speaks about when you talk about that. Coming back to mango malformations, you are looking at the, mango malformation is one of the very severe diseases in case of mango. You have a healthy flower, you have a malformed flower, and you have a malformed flower and healthy flower, all the together spectra is getting different. This is again one of the PhD students who worked with me. And based on that, you can characterize which body is really malformed, which is healthy. And interestingly, again, using red edge, you are able to differentiate that which is healthy, which is malformed. Again, it has a huge applications. The student worked on completely on PhD on this work. What are those areas where the mango malformation is done using hyperspectral imaging data and map them? And coming back to very interesting, like you do finger, what do you call that? Fingerprinting mapping, or what do you do? Like whether that offspring belongs to you or not. Same also, really is the re-increment inbred lines. I have something around 1,000 plus. These are the two parents of the plant, Raj 3765, SD 2329. These are two varieties. They are good attribute with respect to yield and nitrogen use efficiency. I put them together. My purpose is to see if at all I can get a genotype derived out of it, which can be high nitrogen use efficient and better yield. So many times you don't get all those attributes together. So my purpose is I can design a new plant type which can have low nitrogen but higher yield. So these are what different genotypes or reels we try to create, and took their spectral signatures and took the spectral signature of parent. And what you do conventionally is parents' attributes like what is height, what is chlorophyll content, what is nitrogen content, what is the yield, and go for matching with all these offsprings. Now, what we are doing is we are doing from the signal itself, spectral signature, we are trying to match with the offspring or those plants, which is best matching with the parents. That means it has the same attribute what the parent is having. That means it has inherited same attribute what I was looking for. So this is what simply through the spectral matching, that's called gem distance matching, I can differentiate which genotype is really good, which is bad, what I was looking for. Now time has come. You don't need to go for conventional approach, the genetics of breeding. You can do this using hyperspectral remote sensing in the field scale, what we do in our conditions. Again, you can do a lot of discrimination, like there is a lot of challenges with respect to wheat and weed, like crop and wheat. Like you have orchard crops, how you can discriminate. Hyperspectral has a solutions for that. You can do for the weed and plant, and also within the plant, you can do all those kind of discriminations. This is what, again, from the satellite, we try to do. Both multispectral and hyperspectral, we try to compare vis-a-vis multispectral, hyperspectral, how the discrimination capability is better in case of hyperspectral compared to multispectral. This is somewhere in Bhagalpur, where again, we had an airborne campaign. Around 8 crops were there. Same time also, we have a ground measured ASC data, spectroscopy data, and we were able to see the spectral discrimination and classification, how good and accuracy is that. That means it is possible to do that discrimination. And this is what the bigger program we had in country called Chaman for the horticulture crop estimation accuracy. Hyperspectral, wherever you have a challenges, hyperspectral has a solutions for that purpose. So coming back to the solutions, so you have a scanning done by hyperspectral by the drone or from the satellite. You are having a variable color map like this, what you have. And then you can go further. So each pixel speaks about whether you have a nitrogen content, less or more, or water stress or whatever. Then accordingly, you can go for variable input applications. That's what each pixel you can socialize, you can make familiarize for the benefit of the society. This is what the spectroscopy helps to. So this is what I told about the digital soil map. Now this is what I have my field of precision farming field. And these are the maps which I have generated for the nitrogen, phosphorus, potassium, whatever you have. And I have a digital soil map of that same field also. So I click on any pixel, I have all the soil attributes, I have all plant attributes standing over there. So now I need to go do where to do with respect to best health management. So this is what the socializing the pixel. Each pixel, I'm able to characterize, letting know what stress is happening and how management option can be. This is what the precision farming is. So coming back to some of those research issues and challenges, I think many students, they have joined over here. You know, the spectra which you have, it's a composite effect of many attributes. How to characterize, how to understand what are those attributes which is behaving, which is affecting the spectra. That's what the biggest challenge what you have. So that you should discern each part of the spectrum with respect to attribute and characterize those attributes. And that's what, because this is what the challenge is, composite effect you have on the spectra, you have to decompose that. Then coming back to the pest and disease, these are the challenges what we have. Spatial extent of pest and disease is a big challenge. And discrimination of different stresses is a big challenge. Scoring 0 to 9 is a big challenge for us. We need to go for the more quantitative scoring. Severity level assessment, then economic injury level. Unless until I know that, I trigger that, then no management can be triggered for that purpose. Then sensor-based early prediction. This is what with respect to pest and disease. Then you have many sensors, many instruments all around the world. So you should go to inter-sensor calibrations for developing a standardized spectral library. You may have I have a ASD spectrometer, you have a specking spectrometer, somebody has another spectra register, somebody. So the spectra altogether is going to be different. Spectral response is different. Signal noise ratio is different. Or you have an imaging spectrometer, something different, I have different. So that's what the challenge, one has to do that. That's what the spectroscopy community should look for. And most importantly, these are all luxurious research. We look at the spectroscopy, very expensive instruments. I'm coming to the next slide talking on that, starting from the imaging to non-imaging. But from these, we should derive low-cost technologies, like for NIR spectroscopy can be used for many applications. So we can have a low-cost technology for the sucrose, or for the mango ripening, or tomato ripening, or anything, any purpose. So that can be done from this hyperspectral. We can come down. What is the using all the data mining techniques, feature selection techniques for that purpose. So these are some of those data issues. I think I'm not going to read each of them because of the time constraint. And again, there are a lot of challenges you have. When you talk of hyperspectral, always there is a mixed pixel effect, impure pixel always. That's why we call it pure spectra, or pure end member should be always there. Otherwise, the pixel may have a covered by soil and happily covered by water. The spectra is going to be different. So unmixing of the pixel is most challenging. There is again, many times that's a linear and non-linear. One has to address that. Again, I have not put any slides on that. Anybody, we can discuss on that again. And it's a very large data volume. Always you need a high-end computing system. The kind of drone what we are flying, one time flying is 140 GB data we capture. So think of the kind of data storage and also maintaining that data and analyzing that data. And cost, of course, I told about the cost. Data processing chains, and it takes maybe days together also processing this data when you, such a huge data when you capture, and the centimeter level pixel size you have and a huge spectral data. So that's what, and spectral library, we should have a standard spectral library. That's what we are working on. Calibration and validation is most important with respect to before going to take any sensor or sensor evaluations. Then we should go for the combined visible and NIR spectroscopy and also thermal infrared spectroscopy, pulling them together to see which are really opportunities to harvest for that purpose. And it's really a complex data analysis. So major issue is most of the recent hyperspectral research is combined to Hyperion and also recent NMapper and Prisma satellite data and ground-based spectrometry. Proximal hyperspectral remote sensing use ground-based spectrometry has been confined to very few because of non-availability of expensive spectrometers. So proximal thermal hyperspectral, again, hardly anybody is having, so it's not explored much. So except few ground or airborne imaging spectrometry is yet to be explored by the user community. So that's what the major issues or challenges when you talk of the remote sensing community, particularly in India, are the researchers what we have. So we should have a spectral library of hard surface features, irrespective of the domain you look at. That's what our purpose is, both from local to regional to global scale. Globally, there are many countries, they have developed their own spectral library. Now, our emphasis is on doing that only. Understanding physical and chemical and biological properties of target and characterization, that is most important. You should have a domain expertise, then only you can understand the spectroscopy. You don't have a domain expertise, you run a machine learning and deep learning, something will come out and you publish the paper, but nothing will go in a scale, an operational scale, and that's what the challenge is. And many times it happens, you understand that. Monitoring and mapping and discrimination, this is very, very important with respect to spectroscopy, is a very potential area. Discrimination with respect to crop type, genotypes, stress levels, all those things can be done. You can create a huge thematic products. And understanding different ecological processes, like we are talking of regenerative agriculture, talking of carbon sequestration, talking of like photosynthesis, light dimming and effect on the plant, all those things you can do by this. And so the challenge is there are many opportunities, many approaches. People, you go to each individual, they have their own approach. Many approaches, tools and applications need to be standardized. This is what the challenge we should look for. Local to regional studies and pilot projects need to be a larger scale. Then applications should demand for a new ecosystem products, so that we should go for larger applications to meet the need of the society and better life. So this is what the future thrust, what we are looking at. So we should look for prospective plan of India, hyperspectral remote sensing. That's what we had in Asia program, we had average energy program. And this is what we have fought over the time. And now again, new program we are going to take away soon. And then collaborate efforts and agencies for the hyperspectral sensors. There should be demand to ISRO to run hyperspectral satellite. Till now, we don't have. Whatever we had, that's gone cases. But we don't have, right now we don't have. We are always borrowing from Europe or Prisma and NASA. And possible to look for it, work on thermal hyperspectral remote sensing. It has a huge applications. You look at the pollutions, what is happening in the industry. Go to the rooftop, put a hyperspectral teleps camera, thermal hyperspectral, and just scan through. Within no time, you know which industry is really polluting, which is not. You scan through a playground of football match is going, and you can know who is cluster is really drunk, which is not, who is smoking, who is not smoking. So it has a huge applications you look for. So, and also light conditions, like light, like in a city in the night, you fly the hyperspectral in camera, and you can know how efficient is your city management with respect to the energy, because a different light has different energy consumptions. This is what the huge future applications people have been looking for. Because of time constraint, I didn't cover the geology, water. There are many, because we had 37 centers working on different parts. But this is what, in brief, I wanted to tell. And this is not my individual contribution. It's a huge team involved and over the years. And I must thank each and every one. My students, my research fellows, my gurus like Bandhavada Sahab, and fellow colleagues like Rajiv Sahab, who is associated with us on the NIR Spectroscopy for the sucrose. Thank you. Thank you very much. I'll be happy to take any questions or any discussion. I have to do that. Just a minute.
Yeah, thank you very much, Dr. Sahu, for your excellent presentation. And I know you very well for the last maybe more than 15 years, that you have worked extensively on multispectral, hyperspectral applications, and particularly on agriculture. And you have shown the area of work that you have undertaken with so many collaborators, not only in India, but also from outside India. And I think there may be many questions to you. I don't know how much time you have, but any questions from the students and other researchers who are present here? And I think I will have some more questions from here, those who are present. Okay, let me start first. I have some questions. The first question is that in most of the data that we have shown, the R square value, particularly for satellite images, is not really very high. It's really roughly around 0.5 or so. So will that be acceptable, or what is your view on that? How can that be improved, or with the data we can continue?
Answer is there are many studies with R square 0.5, 5.6 also become significant and acceptable. That's what the publications will take off. One second is this again, there is a always open opportunity to improve further. The challenge with the satellite data is noises, the bands which you remove, and also the soil sampling, the plant sampling which you do, that do from one particular plant or any particular point soil sampling you do. But when you look at the satellite data, that is 30 by 30 feet. So to represent that sampling is the challenge. Of course, we have taken the composite sampling. So this is what the second. And third is the kind of model which is used. So with time, we are trying to improve upon the model. And again, because of time constraint, I have talked about generative AI, which are trying to use to simulate the PEG spectrum, so that you within generative AI. And most of the time, in case of soil, you could see in case of my field, 5-acre field, you can't expect soil variations, but you may have spectral variations maybe because of many other activities. Unless until I go in a large scale for the soil mapping, you can't get a good model. That's what the challenge always remains. So in case of satellite or in case of airborne campaign, what we did, most of the time is a very narrow strip. In case of Hyperion, it was something around 100 by 7 kilometers. That was a good sufficient data set, 82 data points. Variations. But when you look at the airborne, it was a very small field, 40 by 10 kilometers. You can't expect much variations with the soil. So these are some of those challenges. So that's the reason you have a limited soil sample, limited spectrum. So we are trying to do is now we are simulating the spectrum, fake spectrum with simulating. That's what the new research is come up called GAN. Generative Adverse Network is the new concept, which is we are using to simulate those spectra and also simulating corresponding activity and validating that what you have ground measured. Once it's done, like in case of ready to transfer model, I can create a million spectra, and also then I can go for matching and do that. Same also we are trying to do for the soil to bring better accuracy, so that I have a huge spectra with huge variations and go for matching. And this has been done with a very limited study, limited time. So, as I was saying, we have collected something around 7,000 plus spectra right now, and we are trying to improve upon the model to bring the better accuracy.
Yeah. Thank you very much. This is a very good suggestion, because in most of the applications we find that it's very difficult to get data, spectral data and the corresponding the reference values and whatever we have to calibrate. So this type of the spectra.
Another point which he has already told, the research evolutions. When you actually relate which part of the spectrum is because of what, if you can narrow down to that, your relationship will be far better. Now what is happening is you are taking
The
all together.
spectrum. Yes.
So it is masking many relations.
Correct. Unnecessarily,
not only that, there will be a lot of correlations between the reflectance values of different parts of the spectrum. So their autocorrelation will be there. So that will also mar your R-square values and all this. So very important point which I again stress, that you require the biological side of the research much more to use these techniques. These techniques are now quite developed, advanced. But the biological side, in case of our case, maybe physical side you are using for say pollution or something. So this research is much more required than even pinpointer.
No, this is a very good input from you. I mean,
So
we can
that is the key. That the background research to use this tool.
So this is what we do is another point what Sarah was talking. You have a reference. Reference is function of so many attributes. For example, soil or plant. But what I do is I take X attribute as a function of reference. Whereas that reference is affected by number of attributes. So we should do opposite. Rather we don't do many papers you see. So that's what right now we're doing that only. If at all I can rejoin those traces, those spectra, two different attributes. So we have done, Sarah is asking biological studies, again because of time constraint. We have taken a very rich organic carbon content soil. The rice soil we have taken around 2.56 organic carbon content. That we did a chemical treatment of that. That is hydrogen peroxide treatment we did, and we bring down to 0.2. Around 30 samples we have generated out of it and replications. So 3 replications, 30 samples is getting 90 samples. So your spectra, the soil is same, only organic carbon content is changing. Nothing else is changing. I have taken spectra and analyze that, find out what is that sensitivity. So this is what each parameter one has to do. This is what the lab conditions, having understanding the soil chemistry, one has to do, relate which part is really.
No, that you have shown in this chlorophyll example, that only changing the chlorophyll
That's
part.
simulation ID. Same we are doing, we have done also in case of soil.
The real
Doing
sample,
in the soil changes.
with the real samples.
Because no effect of clay, no effect of moisture or any other. Mineral, nothing. Only organic carbon. Same also you can do for nitrogen, phosphorus, sulfur. We are doing that. And they can be spectral library because
Exactly.
each spectrum cannot be a spectral signature. Each spectral signature cannot be spectral library. One has to remember that. One has to standardize what is that data based on this. You tell it is a spectral signature. Otherwise, you can create a huge.
Unless there is a lot of
That
noise.
Yeah. There is a procedure to create a spectral library.
So, in your opinion, in a nutshell, this you mean to say that this hyperspectral has an edge over this multispectral for different applications. Okay.
Whenever, actually, let me tell you, I was talking to a that's called Ramakrishna. He is a good friend of mine. He is working in NASA. He was a high alumni for a few months, then he left NASA. So we had a meeting with him, discussing Washington DC. So I was asking why NASA is not taking up any initiative with respect to hyperspectral sensor hyperspectral. They say if you look at the economics and if you optimize the trade-off, multispectral vis-a-vis hyperspectral and the benefit out of it, multispectral is good enough in the sense that you need yield estimation, you need a crop yield acreage estimation or something like that. That much band is enough. The 4 bands are enough for meeting around 70% of my work. Only for 30% why I should spend money. And the US was that time under financial recession. So it says there is a mission, big mission from history. I was part of that. That took a backstage. That's the reason why I asked. The Senate has not approved that to go because of this economics. So it's a luxury situation. Same also in Canada. They say it's a luxury situation. Wherever multispectral fails, whatever you have a challenge, there you should go for hyperspectral. Like spectral camouflaging, or business applications. Many applications, like you perspective of getting the diamonds, golds, mineralogical mapping, water, all those compositions monitoring, which using only multispectral you are not able to do. So hyperspectral is important.
Okay. Thank you. We will definitely continue. Any other questions? Professor Shan Chatterjee is from our electronics department.
I have some queries related to some plant. Actually, I have a project on mangrove classification. And here we have collected samples from water, salinity to measure salinity, the soil in the Sundarban area, and the mangrove species, and some photos. So the question is that how I can use the spectral characterization process for this soil first, then maybe water, because our need of the project is that in Sundarban area, the sweet mangroves are almost completely gone, and the saline mangroves are in a big manner, and many area almost no mangroves, and that creates a huge problem in the cyclone path. So how I can have this process in my lab, or what are the process I can use in that case?
So what is that you have measuring instruments?
We have our environmental lab in civil engineering department, and the instrument they are using, those are chemical based. So that's the problem. So that's why I am thinking that whether I don't know, Rajiv sir's spectral characterization, can it be helped to get anything from the soil? I think no.
The soil you can do. Soil calibration can be done. But the question which you asked, that you just can have an understanding and characterization of those ecosystem you can do by using spectroscopy. But solution somebody has to come up.
Yes, solutions has to come up, because you cannot get idea that which plant, because our process is that where I should plant a particular tree, whether that soil is at all taking that kind of tree.
Okay.
So that
So first part is mangrove estimation classification.
that's the first part.
That part can be done very easily using hyperspectral.
Okay.
Many people have done that to Sundarban area also.
Okay.
And she's here, she's a professor in engineering college. She is part of my dissertation
Where in Yadupur University?
in
Oh.
Kolkata itself. I can see her name and
Okay.
she was our team working on hyperspectral. So
Okay.
that would be the team.
Okay.
And I gave some online lectures there for the engineering college. That was my college, now it became a university.
Shibpur.
No.
I see Shibpur. No.
I'll let you know.
JIS.
So they were they did done mangrove. You have a ISRO center here also, they have done mangrove.
Okay.
Using airborne data, mangrove calculations. Now we have the second part is if you can do the soil sample analysis and do the characterization with respect to the attribute report, there is a suitability analysis. Which plant or which crop can be grown which area based on the soil attribute.
Yes.
That is standard. There is no science here.
Right.
You can do that. That is available. NPSS LP is the source contact point. There are publications, there is a report available. There is a categories N1, N2, N2, non-suitable, S1, S2, S3 is suitability category ranking. Based on that soil attribute, you can go for the suitability analysis and go for the what is the existing land use you can do from the image. What would be optimum land use based on this soil analysis planning that you can do.
Okay. Another question is that you said that drone-based spectroscopy. So, what I have been, as I am in our lab department, have a IoT lab. So we initially thought of a smart agriculture project. Now, drone-based smart agriculture project we initially take care. Then what we saw that to get precision image, drone may be lowered in height, and in that case, the air thrust in the lower bottom portion of the drone creates a problem in the actual image, because the crops may have some air problem on its side. So how in that case I can get the right kind of image?
And what height is that?
It's not very high, because it's not hyperspectral image. It's just a normal visual image.
At what height you are?
It's basically 10 meter, 20 meter. I think that's
10 meter. Could you do the image processing also?
It's done in the laboratory. What they have done, they have taken in they call Bardhaman region on a paddy field, and they got to 20 meter height, and they take the
20 meters is okay. 10 meters.
10 meter probably नहीं होगा 20 मीटर से ऑटो आ रहा है थोड़ा थोड़ा आ रहा था
That's okay.
उसमें कुछ प्रॉब्लम नहीं होगा
In fact, we need to try around 3 meter height for the staying.
Yes, yeah, that's a big problem because when you
that will be a lot of wind source.
yes, lot of winds.
But that helps staying to be done in all possible directions of the canopy.
Okay.
But in case of remote sensing, 20 meter
Yes,
height,
that's
that doesn't. Again, you should take sometimes there is no weight. That should be sufficient height. There is a SOP for the drone remote sensing. That is
Okay, I think any other questions from any other student or scientist who are present here? Anyway, you can definitely you have some question. Okay, Dilip.
Sir, you have done the wavelength selection method, cars and other things. So in that case, you get some specific wavelength. So before doing that, do you have done some dimensional reduction technique, or you just take that full wavelength, then you use that wavelength selection method?
there are two ideas. You can do dimensional reductions. Dimensional reductions again, how do you
That will be reduced. Okay. Then the number of wavelength will be reduced in that case. So
since from the guy you know the specific band, why actually you need to go for the dimensional reduction? You want to get the features.
Yes, yes.
To get that feature. Once I got the feature, then it's only I can just another is you know PC analysis. Which bands are really heavyweight, this particular, there are different approaches. PC is gone. Nowadays, last year using EIF using. There are many techniques. PLS are also weight after putting. Based on that, you can.
and you have done the spectral distance. What do you mean by spectral distance? You have calculated.
How spectrally
You have mentioned about this. I've noted down this Jeffrey's
GM distance.
Yeah.
That is called GM distance.
We have not tried that.
How spectrally
Yeah. That is another very interesting input. We'll try that. We have not used this.
that is spectrally how they are different. So GM distance is example which size the scientists who gave that distance. There's no
You note that JM distance. Shisha, please note that. This will be very useful. Actually, Bangabata sir, he has given one problem to find out the distance between the two types of spectra. In terms of spectra for Lagrangian, then we have one project. So we'll okay, I think any questions from any other scientists, or we'll close the session. Dr. Sahoo is with us for almost 1 hour 45 minutes. So he needs some break also. So in case of you need any assistance or any help or any guidance, I think he's always with us. So you can directly mail him, or you can send any mail to me. I'll direct that mail to you. So on behalf of this Jadavpur University and our Asian Near Trade Consortium Conference Committee, we sincerely thank Dr. Sahoo to come all the way to Jadavpur University physically, and he has interacted with some of the professors here and students. And we'll be very glad if we get continuous support from Dr. Sahoo to make this conference a great success, because he has got wide contact. He is working with so many research groups on spectroscopy in India. So I request Dr. Sahoo to please help us so that we can organize the conference. That conference is, although its focus is on NIR spectroscopy, but we'll also accept papers on hyperspectral, Raman, and other type of spectroscopic techniques in this conference. So the conference date is 8 to 10 December in Kolkata, it's available in our website. All the details are available in the website. In case of any other requirement, you may please contact us. So with that, we conclude our session of this webinar series today. Thank you very much for joining. Thank you very much.
Thank you. Thank you, Rajiv sir, and thank you, your team and organizer. And I look forward to get associated with you and your team. Thank you. Okay. Let's give a big hand. Thank you.